Welcome to Shri Ram Medical College. Today we will explore importance of cytopathology in early cancer diagnosis. Individual cell research is called cytology, and the study of abnormal or diseased cells is called cytopathology. A patient's tissue or fluid sample is applied to a slide and stained. The anatomical pathologist studies the slide under a microscope to identify the number of cells, their types, grouping patterns, and other characteristics, shape, size, nucleus, etc. Finding out whether a condition is present and what the most likely diagnosis is using this information is helpful. The most common application of cytology is as a screening method to detect disease and determine whether further testing is necessary. The examination of a breast lump is an example of screening. A needle aspirate of the lump submitted for cytology, along with the clinician's assessment and imaging testing, will reveal if the breast cells are suspicious for cancer or appear bland/benign. If they look suspect, a bigger needle may be used to extract more tissue and make a diagnosis before surgery, local removal of the lump or removal of the whole breast. Cells that are shed from body surfaces are examined using exfoliative cytology. The cells that line the bladder and the uterine cervix, the mouth of the womb, are two examples. A minimally invasive treatment known as a cervical or papinicolau smear is used to analyze cells from the cervix, pap smear. In order to view the cervix up close, the clinician must put a speculum into the vagina. After gently scraping the cervix, cervical cells are extracted, spread at the bedside straight onto glass slides, and sent to a lab for analysis. It is also possible to do a direct examination of the cervical scraped material for the Wart virus, human papillomavirus, one of the main causes of cervical cancer. Aspiration cytology is the examination of cells taken from a mass or organ. This calls for a more intrusive sampling technique known as fine needle aspiration, FNAR. A needle is injected into the body part being investigated, occasionally with imaging, such as an ultrasound or CT scan, to make sure the suspect area is being sampled. If a deep organ or tissue is involved, this treatment may be carried out under minimal sedation or even after local anesthetic injection to numb the skin. Similar to how the cervical smear is created, the recovered cells are expressed onto a slide. When fluid is aspirated, such as from a thyroid cyst, it may be spun in a centrifuge to collect cell-containing particles for study. Urine, sputum, and cervical smears are some of the most often used exfoliative samples in cytology. These are often checked for any questionable cells by qualified cytotechnicians or, in certain labs, computerized automated methods. A pathologist receives suspicious samples for additional microscopic analysis and final diagnosis. Typically, a pathologist will examine the aspirated material immediately. Join Shri Ram Medical College and take the first step toward a successful career in medical laboratory technology. We are here to assist you in launching your career as a medical lab technician. Call us at 9968193009 for more information about our program or to submit an application right away. Shri Ram Medical College, nurturing next generation of healthcare leaders, and offers all paramedical courses at competitive fee and helps in 100% placement assistance. Admission open. For more information, please visit our website ShriRamCollege.com and like, share, subscribe our YouTube channel Shri Ram Medical College. Good luck, goodbye.